Okay, Unit 2 SOL review, still from Pompano Beach. So first thing we got to talk about is pH scale. This is going to be basically the potential of hydrogen ions. This is the highest potential. This is the lowest potential. Each time we go from one number to another number, the number increases or decreases by 10. So this is 10 times more basic than 0. This is 100 times more basic than 0, or basic than 0. This is 10,000 times more basic than 0, because this is the acidic side. This is the basic side, so if we move this way we get more basic. If we move this way we get more acidic. And right in the middle, 7, we have what's called neutral. Put another perspective, we got battery acid at the acidic end, so it's increasing acidity going this way. We have lye, which is used in soap, for the alkaline end or the basic end, and then increasing alkalinity this way. You can see milk is somewhere almost right in the middle, normal stream water right in the middle, seawater is a little bit basic, normal rain or precipitation is a little acidic. Fish production is affected. Adult fish die if we get to about a pH of 3.5 or more acidic. We have vinegar, lemon juice, and battery acid. Acid rain can actually be in a whole range of things. Then we have baking soda, milk of magnesia, ammonia, and lye. So if we put 14 in with 0, we get 7. And then the other thing we had to do is we had to come up with reactions and products, and I think it was peanut butter and jelly and bread, making a peanut butter and jelly sandwich. Here we have a girl sitting on a spare tire. We have a flat tire. She's got a lug wrench. She's got a lift. Looks like another lug wrench right there as well. So if we take a lug wrench, we take elbow grease, a car jack, we take a spare tire, we actually get smiles and miles. So basically this side is the reactants, this side is the products, and we get a fixed tire from a flat tire. Then we have the CHNOPs, the 5 or 6 most prevalent atoms in biological cells and activities. We talked about them as being hydrocarbons. This actually makes the organic section. We put in O, we can start making carbohydrates and lipids, which are two of the basic materials needed for life. We throw in nitrogen, we can get some amino acids and proteins. We throw in sulfur, we get more amino acids and proteins. And if we throw in phosphorus, we get the nucleic acids like RNA and DNA. Then we also need some other elements for some of the other proteins, because proteins, remember, make us, and we need as many things as we can to make the proteins we need. Then we also, there was supposed to be a mnemonic, which I had here, and I hated it, so I got rid of it, but you're supposed to come up with a mnemonic to remember these, and you can come up with something for the C, the H, the O, the N, the P, and the S, and this is basically the importance in terms of the biological materials. Then we have the water molecule, which is a Mickey Mouse molecule, made of 2 hydrogens and an oxygen. The electrons spend more time around the oxygen, and it's a covalent bond, so we actually send one oxygen around hydrogen thinking it's got 2, and another one 2, and then the 2 hydrogens go with an 8 to make a 10. So we actually make a very covalent bonded into basically a noble molecule. This one actually is polar. We've got a plus end and a negative end, and because of that the water actually aligns so that the plus end of the hydrogen sticks with the negative end of the oxygen, and we have this. I think you're supposed to actually put in 5 water molecules and draw dotted lines for the hydrogen bonding that takes place between the water molecules that allow us to actually have surface tension. Then we're talking about cohesion and adhesion. Cohesion actually making the water droplets when the water molecules stick with water molecules, and adhesion is when the water molecules stick to the spider web or anything else. The cohesion is actually very strong on the inside, but it's even stronger on the outside because the outside molecules have no more water to latch on, and so they actually spend more of their effort with the molecule right beside it. Then we have the ability of water to cling to, and actually climb material called capillary action. Cohesion of water, allows the molecules, which are held together very strongly at the surface of the water, allows other critters to actually walk on water. Water drop caused by surface tension, where the outside is actually grabbing on to the outside stronger than they are the inside. We have the latent heat of water, where it actually takes energy to cause the ice to start to move enough so that it starts to melt. It takes 80 calories per gram. And then we also have the liquid water moving into steam. That takes 540 calories per gram. It takes more energy to turn water into vapor than it does from ice into water. And then the same thing, we get a cooling process when the vapor turns back into liquid, and we get actually it warms up the atmosphere, but it cools the water, warms up the atmosphere of the environment, and then we have another warming right there because we're actually taking the heat out of the water to turn it into ice. And then the same thing, this is the latent heat, so the solid actually increases in temperature, and then as it melts, gets to that point, it actually settles all out. And then of course it actually changes into a liquid and then goes up and then does the same thing from a liquid to a gas. It's got the hidden heat section in it. It also has the ability to latch on to, in this case, salt, the sodium and chloride, and it actually goes around, so the plus goes around the chlorine ion, which is negative, and the negative end goes to the sodium ion, which is positive, and it rips the two apart, making salty water. We also have diffusion, where we put colored water into water, and we actually watch it diffuse through and eventually cover the whole container. And then we have properties of water chart, where you're supposed to talk about capillary action. Capillary action is the actual water rising up in a tube against the water gradient. And it happens in terms of roots taking in the water, and then it actually pushes the water up through the stem, through a material called xylem, and then it actually goes out to the leaf, and then goes through the stoma, which is just an opening, and actually goes out to the atmosphere. And then more water comes up to replace it. And this is why plants need the water. We need water to go here to power this whole process. If not, the leaves wilt, the stems wilt, and the tree dies. Surface tension, where water actually can form a drop. It can also cohese to anything else, but it cuts the drop of the water, which is surface tension. Resistance to temperature change. We can throw basically, this is whale blubber, but it's actually made of lipids with lots of water inside of it. But it basically keeps the whale nice and toasty, and of course, unless somebody strips the blubber away from the whale. Universal solvent. This is an oceanic process. This is called the calcium carbonate, CCD, calcium carbonate. Oh, I can't remember that. It'll come compensation depth. Calcium carbonate compensation depth. And basically what happens is up at the surface we get calcium forming, and it rains down, and if we have a part of the ocean that's tall enough, it'll actually form on here, a white crystal, almost looks like marine snow. And then if it gets deeper than that, the water gets too cold, and the calcium carbonate dissolves in the water, and then doesn't really form anymore, and then the siliceous sediments come down on top of it. So really deep water you find siliceous sediments, and really shallower water you find the calcium carbonate, which is why we find coral reefs really close to the surface, and some deep water coral reefs, but we don't find coral reefs down at the bottom of the major ocean. Then we have the expansion of water. You can see water has a size, and as it gets warmer it increases in size, and at 4 degrees it is maximum size. But even when it's ice, it's larger than it is when it's liquid. So this is why ice floats on top of water, especially if the water is 4 degrees, where the water is most dense. And then we have diffusion. So we have a container that's holding all the material. We have the container, and then we let the stuff out, and the material moves out till we basically get the same amount of density, same material outside than inside, and then eventually it'll all level out. Then we talked about monomers. Monomers are the basic makeup of all biological molecules. We call them polymers when they're put together. We take a polymer, we break them apart with hydrolysis. We put monomers together with a process called dehydration synthesis. So small molecules with hydroxyl and hydrogen, we actually take the water out of it, we take this section out, and we can make larger molecules through a process of dehydrogen synthesis. Then we can take large molecules like things we eat for our meals, which have an extra water, and we take that water out, and we actually we start breaking them apart through a process called hydrolysis, and the hydrolysis moving this way, and dehydration synthesis moving this way, making smaller things into larger things, and then take the larger thing this way with hydrolysis making it into the smaller things. Then there's the molecular chart. Carbohydrates, lipids, proteins, and nucleic acids. Carbohydrates, proteins, lipids, and nucleic acids. Carbohydrates, proteins, lipids, nucleic acids, carbohydrates, lipids, proteins. So not quite the same order, but basically what we've got is we've got these four things making up all life. Carbohydrates, the polymers and the monomers. Polymers are glycogen, cellulose, that it makes up bread, and the monomers are the sugars that make it up, like glucose, fructose, galactose, and dextrose. Proteins, polypeptide bonds, made up of amino acids, the monomers. Lots of them, 20 of them are needed for life as human life. Lipids, which are the fats, triglycerides, I'm not sure why that's not coming out, and they're basically oils and waxes, and the monomer, the triglycerides, which are the fatty acids, the glycols and the fatty acids put together, and we put those together to make oils and waxes. Oils, the unsaturated, and waxes, saturated fat. And then DNA, deoxyribonucleic acid, ribonucleic acid, the polymers, and then the monomers are the adenosine, guanine, thymine, cytosine, or adenosine, guanine, uracil, or cytosine, which makes up the DNA and the RNA, and what makes up life. And where do we get them? We get them from foods. We can get proteins, we can get fats, we can get sugars, all from our nice complete diet. Don't want to do all veggies unless you make sure you get all the amino acids you need, and you don't want to do all proteins because you're not getting some of the other things that you need for life. Last thing is enzymes. Enzyme is a special protein ending with ASE, usually like sucrase, and it actually is going to take sucrose, put it down, and actually break it into its two monomers, glucose and fructose. So we actually put in water to break it apart. Remember, we hydrolysis to break it apart. And what we basically get is we get a substrate entering the enzyme. This is the enzyme. This is the substrate. It enters at something called the active site. It's a lock and key. So the substrate's got to fit a very specific shaped protein. And remember, proteins are ribbons with very specific shapes. We actually get the enzyme's substrate complex. When we get them together, then we actually put a water molecule into it. We break these two things apart. Now it's called an enzyme product complex. And what happens is the products go out, and the enzyme is there to get the next substrate and just keep going. Remember, enzymes work on only one substrate. We have an optimum temperature where the rate of enzymatic process actually increases and increases to the optimum, and then it decreases. So we actually say that the optimum temperature, this is 40 degrees, and that's the best temperature. And I put the word best in there because I can't put optimum because it's there, but they basically mean the same thing. So it's the optimum temperature to ensure max speed of an enzymatic catalyzed process. And then we can have the same thing with pH. And then we can have some pHs with very acidic. Pepsin works in your stomach. And then we have trypsin actually working in your mouth. And you can, your mouth is a little bit basic, and your stomach is very acidic. and this is the most optimum or the best pH value to ensure the maximum speed of enzymatic processes. And just like different processes, different proteins, different enzymatic reactions happen at different temperatures, so does different pHs. They actually have different enzymatic processes also. This really should be a little bit less than that, because around 38, 39 is actually human body temperature, and that's when most of the enzymatic processes in our body are optimum because of our body temperature. Okay, I think that's it. Thanks for stopping by. That's 2.